we can start. Let me.
Okay. Hello, Dr. Kumar. Hello, Dr. Kumar. Yes, hello.
Hi there.
Can you hear me?
Yes, I can hear you.
Okay. Okay. Hello, everyone. I'm very pleased to welcome all of you to this webinar. And this is the second webinar organized by the Section E2 of the Sri Lanka Association for the Advancement of Science. And I can see a lot of participants today, and thank you for being with us today. And so in the first place, I would like to get your attention for few terms and conditions about this webinar. And usually at the entry and throughout the lecture, your microphones will be muted and your what do you call, the videos will be switched off. This is to avoid any inconvenience to the speaker during his speech. But however, at the end, there is a question and answer session, so you can get interacted with the lecturer during that session, and also you can post your questions in the chat box. And when it comes to Sri Lanka Association for the Advancement of Science, this is the premier organization of science in Sri Lanka. And so this year, the theme of the SLAAS is Science and Technology for Better Tomorrow. In today's presentation, we will realize how nanoscience and nanotechnology have enabled innovative approaches for addressing three main global challenges, namely medical diagnosis and therapy, energy, and inequality. So without further delay, it's my pleasure to introduce our speaker, Dr. Kumar Challa. He is the managing director at the Harvard University and also the former director of the nanotechnology at the Center for Advanced Microstructures and Devices, named as CAMD, at Louisiana State University. He is the president of the two biotech ventures, Xheme Inc. and Quantum Technology Group in Boston. He is involved in the strategy management, technology development, and commercialization of the next generation plastics and quantum dots for biomedical diagnostics. He has more than 80 scientific publications, edited over 40 books, and 2 journals in the field of nanotechnology. And he has been involved with the launch of 3 successful startups. And very importantly, he is my postdoctoral advisor. And I learned nanotechnology under his supervision. And he has always been very instrumental in my career. And so, Dr. Kumar, over to you. Thank you very much.
Okay, good evening, everyone. Yeah, first of all, thank you so much, Rohini, for this wonderful opportunity to interact with you. It's been a while. And also I consider it as a privilege to share my thoughts to wonderful scientists, upcoming scientists, as well as the current scientists, as well as the future scientists that took time from their busy schedules to be here to listen to me. So I would say it's a great honor. Hopefully you will take something, from this 1 hour, 1.5 hours of interaction. And I'm pretty sure I'm going to gain a lot from this as well. I'm also actually, excited to look at the theme for this whole schedule of talks, Better for Tomorrow, right? Better Tomorrow. That's a wonderful and inspiring way to look at how we do science. Always approaching what we do with the intention of making our tomorrow better. So it's very, very exciting to me personally. So as the title for my talk goes today, I want to give you a flavor for what nanoscience and nanotechnology can do in addressing some of the major global challenges. I want to provide snippets of examples from my own research to make this bit more authentic and give you confidence that these tools can indeed be applied to address global challenges. I also want to say at the very outset that I am not the managing director at the Energy Frontier Research Center. I'm on an academic break. I'm focusing on the few startups that I'm involved with. So you will see a flavor of a combination of academic and a couple of examples of industrial research. Again, everything connected to the nanoscience and nanotechnology. So with that, let me start with very first slide. This slide, as you can see, provide you an overview of some of the really major challenges that all of us are facing, no matter where we live, which part of the world we live. These are some of the challenges. And I think as a scientist, and especially those who are interested in nanoscience and nanotechnology, it's always important to consider what is the big problem that we are addressing. Doesn't matter, we could be doing a small reaction in the lab. We could be preparing a very simple nanoparticles in our lab, but always connected to, hey, how is this helping in addressing any one of these major global challenges? So just to give you an idea as to the gravity of these challenges, for example, the energy, right? So it is predicted that by 2050, the energy requirement for maintaining the existing living standards is going to go up by 50%. And that's a huge challenge. So how do you make sure that we not only meet that challenge, but also meet that challenge in such a way that we take care of the environment, we take care of our surroundings, we take care of the lifestyle that we are used to, right? So that's a big challenge. And connected to that is the environmental challenge, right? The best way to understand that is to look at the way the global greenhouse emissions are increasing day by day. And if we don't address that, that leads to cascading negative effects in the way we live today. Water, it's I would say even a more urgent need to address that water challenge, considering that even today, about 200 million gallons of water is being supplied only by several people literally walking to the source of the water to get that water, right? And that's really sad. Now, by 2050, it is also projected that 1 in 4 in the global population will have problems in accessing drinking water. And that's a huge challenge. Coming back to the disease, I don't have to say much about it. We are experiencing a pandemic, right? And how do you deal with these anticipated as well as unanticipated diseases that are going to be coming in our way? That's a major challenge, right? And then, of course, there are other challenges like food, increase in poverty, and so on. I mean, the main idea in making all of us aware of these global challenges is to see what can we do as a nanoscience and nanotechnology practitioners in at least doing our wee bit in addressing these global challenges. For me, in addition to those challenges that we can easily gauge very clearly, and all of us can agree on to, but I think even more bigger challenge is the inequality that is facing the world today, right? There is a wealth disparity. The top 1% of the global population holds majority of the wealth in the world. right? So there is also a gender inequality, right? Women earn much less than men today for the same amount of work. And similarly, the Black people, the Latina women earn even less than the others, right? So there is a severe gender inequality. And there is a racial divide that is dividing all of us today, right? So this is a bigger challenge. If we don't address this, we will pretty much will not see the world that is today in 50 to 100 years down the lane, right? So this is again a big challenge. And I believe the nanoscience, the way we practice nanoscience and nanotechnology will help us to deal with this as well. And this inequality is also seen in terms of the scientific research output, globally speaking. And what you are seeing in this slide is a global map that is more tilted towards the north versus south. That tells us that there is more scientific research output. And obviously that's based on the more ability to carry out that research. It's in the upper northern region of the world, of the globe, than the southern region. So the poverty, that is what is responsible for this inequality in the scientific research output. I think this is something all of us should take note of. So having given you some of the major challenges, let me start with the last one. I wanted to talk about the 3 global challenges, and from my own perspective and from my own experience. The 3 global challenges I want to talk to you all about are the inequality, and then I would talk to you about the biomedical challenge. And then finally, I also want to share with you the global energy challenge and how do we overcome that using the nanotechnology tool. So let's start with a very simple one to understand the inequality challenge, right? So how do we address this inequality challenge? Again, remember, we are wearing the hats of a nanoscience and nanotechnology practitioners. And we all know that to be successful in developing some cool nanoscience and nanotechnologies, it's important to collaborate. Because the nanoscience and nanotechnology is a multidisciplinary, interdisciplinary field that requires partnerships, collaborations, not only within the universities, within the countries, but it requires global partnerships. And I believe such global partnerships will enable us to reduce inequality. And second great example that I have for you is always asking ourselves the question, what can we do with what I have? What can I do with what I have, right? And we have examples of wonderful scientists that demonstrated through their own life struggles, right? Some of these examples that I've given, I'm showing you on this slide, are the ones literally were born into poverty, grew up in poverty, and then kept asking themselves, what can I do with what I have? And then they reach the highest levels that we can imagine. By reaching those highest levels, they not only transform themselves, but also transform the societies around them. That's a great example of how the science in general, the nanoscience and nanotechnology in particular, could help us to beat or meet or overcome this inequality challenge. So now let me come back to the science, the 2 global challenges that require little bit more intimate relationship between the science and the ability to overcome the challenges, global challenges, right? So I want to share with you, again, distilling some of the work that's been done over the several years in my lab at different places and also in my collaborators' labs. I want to distill out this and give you a perspective as to how some of these exciting concepts, the nanoscience and nanotechnology concepts, can be utilized to meet the biomedical challenges. So the very first one, which actually I'm very fond of, is based on a simple molecule, which I call it as, a molecule of, my interest, molecule on which I've spent quite a lot of time. And this molecule is LHRH, which is the luteinizing hormone and releasing hormone. Basically, you could say that the example that I'm going to share with you is this story of this molecule, LHRH. And how this simple molecule that is actually secreted in our, through the pituitary gland, it is produced by our body, can actually be used to develop what are called as targeted contrast agents. And this group of collaborators that I want to introduce you all to are the wonderful set of collaborators who enable for us to collectively come up with this concept and also demonstrate that such a targeted contrast agent can actually revolutionize the disease detection. The other very important take-home message I want to share with you by introducing you to these wonderful group of collaborators are, they all come from diverse backgrounds. They've all experienced the inequality in their own way, right? They've all overcome those inequalities to come together, right, to solve a problem, a scientific problem that has implication towards meeting the biomedical challenge. And that's really an important message. So let's talk about the science, underlying science. I'm not going to go into all the details of it, but hopefully we'll give you an overview of the concept that we use to demonstrate this. So what we did was we took this LHRH molecule. And why did we take this LHRH molecule? Because the LHRH molecule has receptors on majority of the primary tumors as well as the metastatic tumor cells. So if you look at the top 10 cancers today, so the LHRH has receptors on those primary tumor cells as well as the metastatic tumor cells. So if we can bind a magnetic nanoparticles, a very simple iron oxide nanoparticle, it doesn't have to be anything fancy, right? All of us are familiar with the iron oxide nanoparticles. We are all familiar with the rust, right? So basically it's a very simple iron oxide nanoparticle, but it has excellent magnetic properties to be utilized as magnetic resonance imaging contrast agent. So our hypothesis was if you can bind an iron oxide nanoparticle to the LHRH, then we could target majority of the primary tumors as well as the metastatic tumors. Now our interest is more focused towards the metastatic tumor cell detection simply because the way you can detect these primary tumors, it's very well established. And there are also successful therapeutic approaches to deal with the primary tumors compared to the metastatic tumor cells. So if you can, there are less and insensitive techniques that are there to detect the metastatic tumor cells, and there is a major need to address that challenge. So our hypothesis was that if you could bind the simple iron oxide nanoparticle to the LHRH, it would go and target the metastatic tumor cells of the primary tumors in several tissue cells. And primarily we were focusing on the lungs. So what we did was we injected this. I'm not going to the details. There are a lot of science involved in it, and then it has taken us several years to standardize these methodologies. But what we did was, simply speaking, we injected this, and then we observed that these contrast agents targeted specifically the lung metastatic tumor cells. And then we could image them more successfully using what is called as hyperpolarized helium-based MRI technique. So when we did that, what we have seen is, this is the sum and substance of what our discovery was. If you are interested, you can read all the details in one of the proceedings of the National Academy of Science publication. But the sum and substance of this is, this really enabled us to image lung metastatic tumor cell, which is about 400 millimeters, right? So this is the smallest metastatic tumor cell dimension that ever imaged so far, right? So that's the beauty of this technique. Now, the other beauty of this technique is that this also potentially could pave way for understanding the molecular interactions that happens in the metastatic tumor cell progression. So one could imagine imaging such progression through molecular imaging of the lung processes. I think that's what is very exciting for us. The result that we have seen so far, the conceptualization of targeted metastatic MRI contrast agents can be understood from the existing technologies that are available today. For example, if you look at the CT, X-ray computed tomography technique, it's the most sensitive technique that available today. You can detect about 1 to 2 millimeter size lesions, but it cannot really differentiate between benign versus cancerous. But on the other hand, you have a wonderful technique such as PET that's extremely sensitive and can differentiate between the cancer lesion versus the benign, but its sensitivity is only about 5 millimeters, right? So if you look at the context of the targeted MRI contrast agent that we developed, you would really appreciate the potential possibilities in overcoming the challenge of detecting metastatic tumor cells as early and as sensitively as possible. Now, coming back to the story of LHRH. Now, this LHRH is such a beautiful molecule. Now you can imagine, instead of binding an iron oxide nanoparticle to LHRH in order to make a targeted magnetic resonance imaging contrast agent, you bind that to a therapeutic drug such as Lytic Peptide. And what you would see is, again, equally impressive therapeutic outcomes. In fact, this is the concept on which there is a company, Esperance Pharmaceuticals, which I had an opportunity to be part of in the initial stages. This drug is in the final clinical stages for several types of metastatic cancers, including the ovarian one. So here the concept is that the LHRH would go and bind to the tumor cell membrane, and the Lytic peptide is introduced into the cell through the mechanism of endocytosis, and thereby destroying very specifically the metastatic tumor cells here. So this is, again, the whole story of LHRH, how it was able to be utilized as a targeting agent for detection of metastatic tumor cells. It can also be utilized as a targeting agent for sensitive delivery of Lytic peptides or cancer drugs into these tumor cells. So now let's move on to the second story. Again, talks about a challenge that exists in the biomedical sciences. The challenge is, how can you deliver a drug in a time-dependent and dose-dependent fashion, right? So I want to introduce, before I go on to describing the conceptual science behind this, I also want to introduce you to some of the grad students, and I want to really dedicate this talk to these graduate students who enabled us to make these advances. these graduate students are wonderful. They're so dedicated, like every graduate student in this world. I think my hats off to all the graduate students who are actually listening to me today. You all need to be really proud of yourself for your dedication, for your passion, and for your effort in your own way in trying to address some of these global challenges. And without the support from the graduate students, none of these major discoveries that are happening around the world would not have happened at all. And these graduate students are again an example to demonstrate how they've overcome the inequality challenges that we already talked about, right? So with that background, let me dive deep into the science as to how we developed these time and dose dependent magnetically controlled drug delivery systems. So when you talk about the drug delivery systems and how do we control them, you can control them by various means. You could control them optically, right? You can control them thermally, you can control them magnetically. And our focus was on utilizing and taking advantage of the unique magnetic properties and manipulating them in order to develop these controlled drug delivery systems. So there is one example which is very fascinating, is about using a simple technique called layer by layer assembly approach, where you can encapsulate magnetic nanoparticles within a shell. So if you can imagine a core shell, an empty core, and a polymer shell nano architecture that you see here. In this particular case, we have taken the cobalt core and gold shell nanoparticles encapsulated in polyelectrolyte layers, right? And then you create an empty core. So you take such constructs and then you incubate them in fluorescent solutions. And when you apply a magnetic field, what happens is the oscillating magnetic field creates the crevices within the polymeric shell and enables the fluorescent solution to enter the core. And what we see as a first proof of concept demonstration is that the oscillating magnetic fields create within the polymeric shell crevices that enables the release of the drug. And this modulation can be time dependent. It can also be dose dependent. And here is an example that's even more complex, at the same time provides a much more flexibility and an opportunity to include not only the delivery system, but a delivery system in conjunction with detection. So sometimes we call this as theranostic approach. So the concept here is, how do you make sure that you design a nano-based system that is stable, that forms a stable dispersion, so that you can inject them, right? You want to make sure that the solutions are injectable to have practical applications. So the concept need to revolve around designing these architectures so that you prepare stable dispersion. That's one thing. Second thing, you prepare these dispersions to make sure that you carry not only the payload, that is drug, but you also carry structural element that enables you to detect or image using multiple options. So what you see here in this particular slide is a gold-silica, what we call it as a magnetic quantum rattle. And the reason why this concept was developed is because if you look at the gold nanoparticles, gold nanomaterials have known to have excellent properties for biomedical applications, both as carriers of drug as well as for imaging. The problem with them is in the biological media, they all agglomerate pretty quickly, right? So the concept is, how do you keep these gold nanoparticles discrete. At the same time, make sure that the optical properties are maintained, right? While we do that, we also want to make sure that the gold nanoparticles are not directly interacting with the biological medium, right? So one way to do that is to create a silica shell around these. Now the silica is again a beautiful material. It's biocompatible, it's biodegradable. So it offers necessary architecture that can be utilized for in vivo experiments, right? So what we have here, if you look at the on the rightmost corner, A, you would see a silica shell. I'm not going to go into the details of the synthesis, but you see a silica shell that is encapsulated with tiny, atomically precise gold clusters, and the core has slightly bigger gold nanoparticles. Now these bigger gold nanoparticles, about 4 to 5 nanometers, carry the drug, and they also provide attractive near-infrared photonics-based imaging, right? But now we look at the two different types of gold architectures. One, the ones that are encapsulated within the silica shell are very tiny, less than 2 nanometers. And the beauty of the size-dependent gold architectures is the moment the gold becomes less than 2 nanometers, as it becomes closer to atomically precise as possible, it starts demonstrating magnetic properties. So now you have in this architecture small gold nanoparticles that are magnetic in nature, and you have slightly bigger nanoparticle that offers the right kind of optical properties for imaging purposes, right? So that's the coolness about this drug. I'm not going to go into the details of how we image, but it is suffice to share that this was demonstrated to be a successful way to treat colorectal cancer. So we not only imaged in vivo the metastatic tumor cells and primary tumors, but we also deliver the drug in treating those tumor cells. So that's a beauty where conceptually one could imagine you could do both a combination of imaging as well as the therapeutic modalities. So it's quite natural to move from two different types of gold nanoparticles that we talked about in order to understand where is the transition happening, right? The gold nanoparticles, as I said, less than 2 nanometers offers us the magnetic properties, right? Bigger than 2 nanometers actually lead to gold nanoparticles that have unique optical properties, right? So how do we control these magnetic nanoparticles? So the best way one could imagine is to see if you can prepare atomically precise gold clusters, right? And then develop an understanding of, can you tweak the magnetic properties of these small gold clusters by increasing or decreasing the number of gold atoms within them? That's exactly what we sought out to understand. And this is what is called as chemically induced magnetism, right? So the magnetism is arising because you have thiol linkages on these small gold clusters, and the interaction of the thiol linkage to the surface of the metal, right, is what is leading to the unique magnetic properties. Therefore, conceptually, two things are enabling the magnetic properties. One is the interaction, the chemical interaction of the thiol moieties on these gold clusters. The second thing is the size of the gold clusters itself, right? So here where we demonstrated that one could manipulate the magnetic properties by controlling both. And here is another example to show how we can actually manipulate, the magnetic properties by using a third approach. A third approach is, if you can imagine creating a gold shell, right? A gold shell, a thin, possibly a mono layer of gold shell, and then you can biofunctionalize this gold shell in order to attach variety of biofunctional groups. Either could be a targeting agent, right? So when we do that, the question is, can we also manipulate the magnetic properties by stepwise surface functionalization? And what we demonstrated was that that is indeed possible. So here, as you increase the steps in biofunctionalization process, and what we have shown is that you can actually change the magnetic properties, and the magnetic properties change from paramagnetism to a complete room temperature magnetism, right? So this is really an astounding way one could imagine now a simple organic chemistry approach could actually lead to generating magnetic nanomaterials. And then one could imagine manipulating the magnetic properties of those nanomaterials. Now you can also imagine the, what I talked about. How does, develop these concepts that spans across variety of disciplines? It requires organic chemistry, it requires inorganic chemistry, right? And then you start exploring further in developing these as either therapeutic models or diagnostic models. Then you start interacting with medical professionals. So there is a broad spectrum of collaboration that's required. Such collaborations really help us to overcome several of the inequality challenges that I talked about. I want us to keep always these important aspects in mind while we also appreciate the exciting science that we are talking about. So the third example I want to share with you, again, overcoming some of the biomedical challenges. And this is an example that comes from, a science that is being, developed into a technology that has commercial implications, right? So this is the company that I founded a couple of years back, and we are developing next generation plastics with regenerative antioxidant properties, right? That is the science part of it. And I also want to introduce you to my collaborators here. Again, you can see these collaborators bring in a breadth of, the scientific disciplines. Again, it's all multidisciplinary approach that is really required. Now, this is an example that tells you the multidisciplinary approach that is required to be successful in the business world as well, right? So what are we trying to do? What we are developing here are a unique class of metal oxide nanoparticles, right? These metal oxide nanoparticles have this unique ability to mimic several enzymes. For example, in this particular, I'm not going to go into the details of the structure for a variety of reasons, including the protection of the IP. But these metal oxide nanoclusters that we are developing seem to mimic, at least 3 different enzymes. One is superoxide dismutase, and the second one is catalase, right? And then there is also a third oxidase, right? So you can imagine this not only acts as a recyclable redox agent, but it also acts as a regenerative antioxidant agent. Again, all these things can be manipulated depending on the environment that we keep these materials in. Okay, so that's the beauty. So what we are trying to do is, can we develop artificial enzymes, right, that have commercial potential, right? So that's the concept that we are working on. And now, where are we applying these concepts, right? So one of the examples that we are looking at is blood storage. Now, just to give you some background as to why this problem is, is a global challenge. Now, we all know we cannot synthesize blood in the laboratory, right? So we have to depend on the donors. Now, the shelf life of blood is limited, right? And especially some of the blood products such as platelets can only survive for 5 to 7 days. So broadly speaking, blood and blood products can only survive between 5 to 42 days. After that, you need to just throw them out from the storage, right? So the question is, are there some creative ways we can develop in order to increase the storage of the blood, right? So one of the reasons why, we have an opportunity is the blood under storage undergoes oxidative and other degradative damage, right? So if there is a way to create a storage device or a blood bag that not only stores the blood, but also offers an opportunity to, create a protection from such degradative changes. So that's the hypothesis on which we are working on, right? And if our hypothesis is correct, right, if we can develop such a blood bag, now you can imagine we can overcome the current shortage of 30 million units. That's a huge shortage, right? And we can also overcome about 18 million units of blood that is being wasted because the blood expiry date of the blood, right? So it's really a big global challenge, and that can potentially be addressed using nanotechnologies. And this is a practical example. It's not from an academic lab. So what we have demonstrated so far looks very promising, right? So if you look at the red blood cells, again, these are the studies based on the red blood. And you could see both from the light micrographs on the top left, and also the hyperspectral images of the stored red blood cells in the bottom left. And if you look at the red blood cell count, the take-home message from this slide is that you can actually increase the shelf life of stored blood by 30 to 50%, right? Which is huge. And that can be translated not only in terms of the financial gain, right? But also in terms of the lives that we can save, right? These lives translates to millions per year. And that's again, I want you to keep bigger challenge, a big global challenge that can be addressed using the nanoscience and nanotechnology tool. So let's now shift gear and move into, hey, how we can address some of the energy challenges using the nanoscience and nanotechnology tool. Again, mind you, these are just few drops in an ocean, and these are coming from my own lab, right? But there are so many even more exciting research discoveries are being made globally. So let's look at the energy challenge. How do you overcome using the nanotechnology? Few examples from my lab. So again, I'm providing a context right? I gave you a context in terms of the energy usage and what we need in 30 years from now, right? We need to increase our energy production by 50%, is the context for using the nanoscience and nanotechnology to solve global problems. The other side of the context I'm giving you here in this slide, you can see how can you reduce our energy usage, right? So there are both, these are the two sides of the same coin. On one side, we can address the global energy challenge by seeing what are the avenues by you can increase the energy production. On the other side, hey, what are the avenues we can reduce the requirement of energy that's being used in our current processes, right? And primarily, we will focus on the chemical industry. And what is really interesting is the chemical collection accounts for 30 to 40% of the worldwide energy use. And it's actually huge, right? And most of this chemical energy use is relying on the use of catalysts. So if there is a way for us to use the nanoscience and nanotechnology to move away from energy intensive catalytic processes to less energy required processes, then you are saving, right? You are minimizing the energy demand that is required for chemical industry. So let's see, what are some of the efforts that have been involved in this direction? So one example, again, it's a concerted effort that I was part of with the Harvard EFRC team. So we developed, as a part of the team, again, mind you, this is a team effort, right? And the team has a whole range of expertise that's coming together. Computational, you have surface science, you have chemical engineering, you have chemistry, right? A broad range of disciplines came together to address this problem in a very unique fashion. And what this team tried to do was to study this in a very fundamental way, right? So develop the atomistic models, right, computationally, and then grow these clusters, porous catalyst clusters with atomic precision using surface science tools, and then also carry out these reactions at a molecular level using the surface science tools, right? And translate this into realistic experimental conditions where using porous catalysts in a laboratory, in a flask, which can further be scaled up to the industrial scale. So this whole concept of designing from very fundamental science all the way to scale up for an industrial scale is the unique concept. And again, without going to the details, the take-home message here is the breadth of expertise that is required, the amount of effort that is required, right, in addressing this global challenge. So what we have demonstrated over a period of, again, 4 or 5 years is that you can actually improve the selectivity of 2 classes of reactions. You can reduce the energy that is necessary in obtaining the product that we want. So that's the take-home message. So the other example that I want to share with you, again, remember, this is a team. This is a team that I was part of when I was at LSU. And again, this team looked at this energy problem, right? Reducing the energy demand in chemical industry by a different approach. And as a part of this approach, we looked at specifically in solving an energy problem that's also contributing to the environmental degradation, right? So it's the problem of petroleum-based plastics, right? All of us are familiar with the PET bottles all of us drink water and throw these bottles away, right? But they are causing immense damage to the environment, right? And also, these PET bottles, the raw material is based on the petroleum, fossil fuels, right? So in every way, this is a great example of how do you accomplish in utilizing green energy processes to come up with materials, right? And those materials in turn help us to minimize the energy demand that's required in the chemical industry. So this is a great example that demonstrates all these elements. And the other dimension to this whole challenge is instead of having a pathway from fossil fuels to energy, both in terms of the energy consumption as well as the energy development, right? Move away from the fossil fuel based approach to biomass based approach, right? And again, there is a simple molecule that demonstrates such concepts here. And this simple molecule is, if you're an organic chemist, you would think, oh my God, what is such a simple molecule? But it has profound implications to it, right? This molecule is FDCA, 2,5-furan dicarboxylic acid. It's the key chemical, right, that's being now utilized for bioplastics. So our goal was, how do you make that key intermediate from biomass, right? So the approach that we took was we would develop a continuous flow system. And then we start with a product that comes from the biomass, which is HMF, right? Hydroxymethyl furfural. I'm sure all of us are familiar with that, right? And develop a continuous process, right? Again, using a combination of fundamental micro and nanotechnologies. And what we came up with was a process that provides 100% selectivity in the conversion of HMF to FDCA, right? And now this process can be translated, scaled up to the industrial scale as a continuous flow process. So again, hopefully I gave you a flavor as to the global energy challenges. And in my own limited experience, what are the ways, creative ways that we can overcome these energy challenges? So yeah, so we can go for a longer time discussing more examples. But just to summarize what we have learned so far, that there are several global challenges, and all those global challenges potentially can be solved creatively using nanoscience and nanotechnology. And I gave you 3 examples of 3 such global challenges, right, that we were able to address to a limited extent, right, in my laboratory, again, through wonderful collaborations, right? The 3 global challenges examples that I gave you were the inequality challenge, right? The energy challenge and the biomedical challenge. Hopefully these concepts, gave you a flavor to, feel passionate about anything that you do using these nanoscience and nanotechnology tools in your own way to help you feel passionate about the small steps that you are taking in addressing some of these challenges. So finally, this is the crux of all that we do, right? All of us, whether we are a fresh researcher, a student, or someone who has spent 30, 40 years in a laboratory, right? But all of us need to have a purpose. And the purpose is always be focused towards some of these. These are the life lessons that enable us to be a good nanotechnologist, that would enable us to be a better scientist, And finally, most importantly, that would enable us to be a better human being, right? So some of these are, from my own perspective, collaborations are key. Work with everyone, right? Not only within the university, not only within your lab, not only within the country, not only, within the neighboring countries, right? Develop these global partnerships and your success will be much more assured, right? And you'll have fun doing it. And again, it's very important to be passionate, right? In whatever little we do in our own way, in our own laboratory, in our own limited surroundings and settings, right? Connect it to the global challenge. I keep repeating this because that's so important for us to motivate ourselves. The second life lesson for me is no matter what we do, apply the scientific rigor, right? It is so important to remove bias in what we do. And we can only do that by constantly applying the rigor, the scientific analysis to make sure that, we are in the right direction. And the third important lesson that I've learned is there are always solutions for every challenge, whether it's a global challenge, whether it's a personal challenge, or whether it's a simple scientific challenge. No matter what the challenge is, there are always solutions. So think outside the box to find that solution. If you think that the solution is not obvious to you, you will find it. I can tell you, okay, based on my own experience of so many years, there are solutions available. You just need to look and find them, right? And ultimately, what is really important for us to be successful is, yeah, it'll be great to have everything that we want, to be successful. But I think what is even more important is asking ourselves, what can I do with what I have? Will really make us to reach the level, the heights, of the example that we talked about, right? About all these brilliant Nobel laureates who overcame those challenges only because they asked themselves at every stage, what can I do with what I have? So with that, yeah, thank you again for this wonderful opportunity. My goal is to provide you, inspiration, provide you flavor as to what are the tools and techniques that the nanoscience and nanotechnology, will help us to solve some of these challenges. Thank you again.
Thank you so much. It's an awesome lecture, Dr. Kumar, and it's very inspiring. And I'm really touched by the approach you have given to eliminate inequality through nanotechnology. And also you have opened many research avenues for us to explore as well. And thank you so much. And now it's a question and time session. And I will give this to Dr. Lytic Peptide. He's the moderator for the question and answer session. Yes, Lytic, over to you.
Thank you very much, madam. Indeed, Professor Challa, that was very inspirational. All the information that you presented is very relevant to what we see around us today. So thank you very much, sir, for that perfect explanation of the power of nanotechnology in terms of answering all the questions that we have. So like Professor Rohini mentioned, now it's time for questions. So we would like to invite the audience to raise their concerns or questions. If you do have any questions, please feel free to unmute yourself and ask questions. If not, you can post your question in the chat option, in the chat box, or else you may raise your hand and we will prompt you for your question. So if there are any questions, please let us know and we can proceed with the Q&A session. And yes, is there anyone with any questions? While you get questions, Professor Challa, I actually have I do have a question. If I may ask a question, sir.
Sure. Yeah, please.
Sir, you spoke about the importance of gold nanoparticles, specifically in biomedical applications. So I have read quite a lot of articles that debate on the safety of different morphological forms of gold nanoparticles. Some say that spherical gold nanoparticles are safer for biomedical applications compared to anisotropic structures, and some say otherwise. So what is your take on it? Do you agree with that statement, or do you think otherwise, sir?
Great question. In fact, this question can be actually looked at it from a higher perspective that brings you to the two aspects of the nanomaterial. Right? So the best example that I can think of is, I'm sure all of us know Jekyll and Hyde story, right? So it's the same person exhibits two different opposing characteristics. So nanomaterials, I think, can be best explained with that analogy. So anytime we deal with nanomaterial, one needs to keep in mind what is the application, right? What is the environment in which they are being used, right? So depending on these, and what is the type of nanomaterial that's being explored, right? Depending on, say, these factors, these nanomaterials can be either Jekyll or a Hyde, right? So it's very important to have that perspective. So coming back to very specifically to your own question, say, for example, if the agglomeration is a challenge, right? If you're doing your in situ studies, right? But the agglomeration could also be an advantage in some other scenarios, right? So it's very important. The context is so important, and the life cycle understanding of what you are trying to do is equally important.
Yes.
Lyro, can I
Thank you, sir. Yes, sir. Please go ahead. Professor Nalin, please go ahead.
Dr. Kumar, this is Nalin. How are you?
Hey, Nal, good seeing you.
How are you? We have a major problem in this part of the world in terms of getting our research into the commercialization. For example, we are also working on a lot of materials on, I think, the influence by the Rohini working in CAMD 10 years ago, and a lot of work on water purification and drug delivery work and so on. But we find it, even though we have a very good work, very good results, and getting the partnership is a major problem in this part of the world, because of the internal situation is such that there are no investors to take this from lab to commercialization. How do you think that, like the university like you, the big university like Harvard, what advice you can give it to us, or any partners we can find in the future if you have very good work?
Yeah, thank you, Nalin. I think it's a wonderful question. All of us struggle with that, right? There is no easy solution for this challenge, but I think there are ways we can find. There are unique ways that you could imagine, addressing this challenge. Now, number one, I would say is, if you believe that you have a commercializable product, right? The very first thing is, I would say, do a thought experiment. I think all of us can easily do thinking, right? So the thought experiment should really look at the whole life cycle of such commercializable product. So on one side, you would look at the science aspect, right? And on the other side, you also look at the environmental aspect. And the third element that you would look at is, the financial aspect to it, okay? So now let's come back to the financial aspect, which is the most important one. So you would look at, okay, what is the potential financial benefit am I going to get out of it? Is it limited to my region, or is it limited to my country, or is it limited, has a global implications to it, right? So when you do this analysis, it needs to be really thorough. As I said, the rigor not only in the science, but also in the analysis, right? Once you do this analysis and you are still convinced that, this is something that's going to dramatically change our lifestyle over a course of time, I think it's very easy to convince, investors to fund this project. So, so to put it very simply, I think the homework, the groundwork is where I think most of startup ideas fail. So I would say, yeah, do the groundwork.
Okay, thank you very much. Yeah.
Thank you very much, Professor Challa, for that explanation. And so there are a few more questions that has come up in the
I
chat.
have another one.
Please go ahead, Professor Naveen.
Yes.
Yes, Dr. Kumar. Yes, can you hear me, Dr. Kumar?
Yes, yeah, thank you.
Dr. Kumar, it's amazing that we can have these magnetic nanoparticle magnetism from gold clusters. And it's, now what I think was, if you can mix these gold clusters with iron oxide nanoparticles, do you think that magnetism can be improved? Can magnetism be improved?
Great question, Rohini. In fact, the example that I shared with you today, which is iron oxide core, gold shell nanoparticles. Yes. So you would initially start with paramagnetic or super paramagnetic particles, these core shell particles, right? And then as you start functionalizing on the gold surface, as you increase the surface functionalization, you can see that, the magnetism of these core shell architecture changes from super paramagnetism to permanent magnetism. Okay. So yeah, you can actually control the magnetic properties by controlling, the surface functionalization. That's what leads to what is called as chemically induced magnetism.
But
Professor Rohini, are you there?
Okay. Yes. There's a little delay. Okay. Yeah, there's a little delay. Yes, I heard it. Yes, Dr. Kumar?
Yes. Does that make sense?
Yes, definitely. Yes. It's amazing that you have opened a lot of avenues for us to work on as well. Yes, Lahiru, you can go. Go ahead.
Lahiru, I have another question.
Yes, please go ahead.
Not a technical question. Dr. Kumar, again, Nalin. For example, like, you are at Harvard now, and what kind of, this is a administrative type of a question that what are the incentives and the infrastructure or policies for spin-offs from the Harvard scientist? What you have?
Yeah.
What kind of, I don't know whether you can answer that.
Great question. So there are several mechanisms that enable actually, the universities to successfully commercialize technologies coming out of, the university labs, right? So let's look at some of these mechanisms, right? So one mechanism is the policy is that since the university is investing funds in individual laboratories that are developing these technologies, obviously university need to recoup those costs, right? And possibly, be profitable as well, so that you can reinvest that back into, further research. So there is a component of gain for the university, and the percentages vary depending on the university. I've seen these vary from 20% all the way to 60 to 70%. That's held by the university. And then there is a component for the researchers, obviously, right? So that's a policy that you would need to develop that meets your own requirements, but taking into consideration that it involves multiple partners in the commercialization process. The other element is once it goes out of the university, once the university invests in protecting the IP, remember that's also very expensive to protect the IP. So university is not only investing in the foundational, providing the foundational scaffolding in terms of the infrastructure, in terms of investing in the salaries of the faculty, right? So in addition to that, they're also investing in the IP protection, which is expensive, especially when it goes to international filing of the patent applications, right? Now, the other element is, as an investor, if someone is licensing this technology, right? So the university need to have the right kind of policies on one side to encourage the investors or the angel investors or the venture capitalist companies, right? It should be attractive for them to license the technology. It's a very delicate balance, right? On one side, you should make it really attractive to the investor, but on the other side, it's also important that the university takes sufficient financial gain out of this, right? So again, so it's very important to have that balance. That balance needs to be looked at not only from the university policy point of view, but also from the cultural point of view, right? So it's a balance. One needs to think carefully. Again, it's very important that you have administrators that think through these processes carefully, right? Strategy, strategy, strategy. There is no one solution that fits all, right? But the strategic thinking really help you. That's where the leadership comes into play, right? So I would say, yeah, strategic thinking, rigorous thinking, taking into consideration the environments around, and have a policy. Never articulate anything or implement anything without thinking through and developing a policy.
Thank you, Dr. Kumar.
You're welcome.
Lahiru, I think there are questions in the chat box.
Yes, I have been looking at the chat box. There are a few questions that has come up in the chat box. One of the questions that has been raised by one of our participants is, do you have any suggestions for any method for graphene production, which is inexpensive and we can carry out in the laboratory? Sir, do you have any explanation for that, sir?
Yeah, it's a good question. Unfortunately, I've never dealt with graphene, so I won't be able to give you a good answer there.
I understand,
Lahiru,
sir.
Lahiru,
I felt the same when I read the question, but yes, sir.
Lahiru, I have a small question. Yeah. Now, Dr. Kumar, again, how do you, this is a technical question, your biomass energy production against the battery technology at the moment Elon Musk is heavily investing on. How do you see these two hand in hand? There are pros and cons between those two, but the battery technology is going to be the technology for the future. How do you, can you compare these two and give a small answer if it is possible, for you? You understand what I mean? Yeah.
Sure. I think it's a good question, right? What are the, we have different energy choices. So how do you make a choice, right? So the important thing to remember is that these are apples and oranges. So apples have their own unique opportunities. Oranges have their unique opportunities, right? So I think it all boils down to the economics. So for example, where the the biomass conversion makes economic sense, I think that's the route one needs to go through. And where it doesn't make sense, then I think that you start looking at other ways to generate the energy.
Thank you. Lahiru?
Yes, personally. And now we have Dr. Danushika with us. She has raised a question. Danushika, can you hear me?
Yes, Dr. Lahiru. Yes, I can hear you.
Please.
So, yes. Thanks a lot, Professor Challa, Kumar, for your very inspirational talk. So since I'm a person who have a very good, I have worked with inox and nanoparticles from my bachelor's. So I have a very general practical issue. This is related to the solubility of inoxide nanoparticles when it is being dried. So say, for example, I have synthesized the inoxide nanoparticles, and then I have got like a pellet of inoxide nanoparticles, which I have dried after some time. So if I want to reuse it back again, so can you suggest us a good solvent? Like if we don't have a very good coating, which is hydrophilic on its surface, so can you just, is there a way for us to disperse it in a solvent for any practical application? Do you have any idea on that one, sir?
Yeah. Again, it's a very good question. This is a problem that we all face all the time, right? So let me give you an approach that would work rather than being very specific to your case, right? So the approach typically would be, yeah, measure the surface charge. Understand what are the reasons, right? Fundamental reasons why the particles agglomerate, right? So always address any challenge that you face in the lab, right? However simple it is, look at it from a fundamental scientific understanding point of view, and then come up with a solution. So now let's talk about in this particular case, right? So there are several reasons why the agglomeration happens, right? But one of the main reasons is the electrostatic interactions. So first thing I would say is measure the zeta potential, if you can, of these particles that you have developed, the surface charge. So once you understand the surface charge, then you can identify surfactants that counteract the surface charge. And the moment you use those surfactants, you would find very well, beautifully dispersed, nanoparticles. So I would say, yeah, that is the way, approach it in a very fundamental scientific way rather than trial and error based approach.
Okay, thanks a lot, Professor. So I have another question. Say, for example, we are going for parenteral administration of inox and nanoparticles, and if we want to target a specific tissue, so do you think it is the magnet? So if we, so the inox and nanoparticles could be, like targeted by using antibodies or like a polymer coating. So do you think it is the magnetic, the strength of the magnetic force, which is the main determinant for the cell penetration or the cell depth that we can reach with these inox and nanoparticles? The preparation, say the parent, if we are going for the parenteral administration or the intravenous administration, is it the strength of the magnetic field that determine the depth of the penetration of the inox and nanoparticles into the cells?
Yeah, very good question. So the answer is no, because the key element here is the targeting agent. It's the targeting agent that drives, remember, it's like someone carrying, right? So the targeting agent carries your nanoparticle to the site, right?
Right.
And then we are taking advantage of the magnetic properties of the nanoparticle.
Okay.
Once it reaches the targeting site. So how do you take advantage of the magnetic nanoparticle? Determines the the application. So if you are using the magnetic nanoparticle as a contrast agent, MRA contrast agent, then the application is going to be imaging. Or if you're going to use the magnetic nanoparticle architecture in such a way that it can deliver the drug, then the other properties of magnetic nanoparticle play a role. For example, if you have polymer in nanoparticle encapsulated with magnetic nanoparticles and drug, and you want to release the drug from the polymer encapsulation, then you would need to apply oscillating magnetic field, right? So then the ways you approach it is different. So it all depends on what is your goal, and accordingly you can fine tune.
All right. Thanks a lot for your response. Yeah. Okay. Thank you. Appreciate it a lot.
Thank you, Dr. Danushika, for your question.
Can I ask something, Lahiru, again?
Please go ahead, personally. Yes.
I think Dr. Kumar, what Darushi asked was, I think for that, I think we don't have a solution. I hope you understand, because she wanted to know, once it is, in the solid form, whether there is a solvent to make it to a suspension. I think during the synthesis, what you meant was the synthesis, we can use the surfactants based on the surface charge and make the suspension. But once you dry it, I don't think, is that what you meant? Once you dry it, it's not possible to disperse even with sonication sometimes. Yeah. So I think what you said is correct. During the synthesis, if you make make use of the surfactants, you can make the highly dispersed nanoparticles of an oxide, but if you dry it completely and then try to disperse it, you are finding problems, I think. Yeah.
Right. Yeah. So the reason why you would find it problem to redisperse after drying is you have not started with the right surfactant to stabilize it before you made it into powder. So if you use the right surfactant during your synthesis process and stabilize it, then when you redisperse it, it would redisperse really well.
Okay. Lahiru?
Yes, Professor Nalin. Thank you, Professor Nalin, and thank you, Professor Challa, for your explanations. So there's one more question that just came up in the chat option. The question is, when we develop a nanoparticle-based anti-cancer drug molecule by using spion nanoparticles, is it necessary to carry out releasing experiments with nanoparticle drug delivery systems? Is the question clear, sir?
Yes. Yeah. Thank you. I'm actually enjoying these questions. They're wonderful questions. Thank you for bringing these questions up. Of course, yeah, it's very important. If you are looking at controlled drug delivery systems, yes, it's very important to carry out those studies in the lab first, and make sure that the conditions are standardized, and also make sure that anticipate what could happen in vivo. Right? And then accordingly tweak the characteristics of these systems so that you're not surprised when you do these experiments in vivo.
Okay. Thank you, Professor Challa. And I myself have another question just out of curiosity, sir, because you mentioned about these magnetic nanoparticles and how the magnetism could be altered by surface functionalization. So is there any simple explanation as to why the magnetic properties of a nanoparticle would change with surface functionalization, sir?
Yeah, it's a great question. So in fact, it's a study, it's pretty well known, and there are probably hundreds of papers that describes the fundamental understanding of what happens. So a simple way to look at this is most of this chemically induced surface functionalization that leads to magnetism is through bonds such as sulfur or nitrogen onto the surface of the gold. So it is understood that the interaction of the sulfur orbitals with the metallic orbitals that leads to this magnetism within the change in the magnetism within the metallic substrate. That's one explanation. I mean, it's more complex than that. Yeah.
Okay, I'm sure. Thank you very much. I mean, that makes a lot of sense. I was curious when I heard about the phenomena, so I wanted to know more about it. And one more thing is, you have spoken a lot about using gold nanoparticles in biomedical applications. Well, I myself am a firm believer that gold is benign, and you can it's perfectly fine to use gold nanoparticles in vivo applications as well. But we know that gold does undergo reactions in the presence of various biomolecules. So do you think that there is a tendency whatsoever for gold nanoparticles to undergo any sort of chemical reactions within the human body out of what we would expect them to do?
Yeah, the answer is yes. Yeah, that's so well established. Even though gold nanoparticles have wonderful characteristics, but in vivo, it fails to, keep up to its promise because of this interaction, strong interactions with, the protein molecules and all kinds of biomolecules within the body. So that's the reason why I think if you remember the example that I gave you of, Gold-silica rattles,
Yes.
where you take the gold nanoparticles, you preserve them, without any interactions with the bodily fluids, right? All the way till it reaches the surface or the tissue that you're targeting.
So that means that there are ways to control the reactivity of nanoparticles so that we can make sure it's perfectly safe.
The gold nanoparticles inside the silica shell.
Thank you very much, sir. That explains a lot. Are there any other questions? If there is anyone who would be willing to ask any questions, please.
Yes, I have one question.
Please go ahead.
Yes, so do we have any possibility to incorporate these gold particles, especially these heme nanomimics, into a polymer system? Like, the functionality, would it affect if you formulate these particles in a dry fiber formulation? Like I'm thinking about going for reservoir applications, like instead of IV, what if we can give these in a solid state formulation, like as if a reservoir, you place it and then expect sustained release? I'm not sure, would it like be stable in that form?
Again, great question. Yeah, so what, if I understand correctly, is you're looking at oral delivery systems, right?
In this aspect, actually, I'm thinking, what if we electrospin these mimics? Can we electrospin them into dry formulations, dry fiber formulations that would lead to sustained release? So that,
right.
Yeah?
Yeah, the answer is yes. So again, let's look at this in a very conceptual fashion, right? So what you're trying to do is to prepare a polymer nanocomposite, right? Polymer gold nanocomposite. Am I correct?
Yes, yes.
Okay, so yes, you can prepare a polymer nanocomposite in a really controlled fashion. And depending on the type of polymer, actually, you can also enable control release if that's what you want to do. Or you can also make sure that the gold nanoparticles don't leach if that's your goal. So you can do both ways. There are avenues to do that. So if you are looking at a controlled release from these composites, there are several ways you can do that. You can select a polymer characteristic such way that it gets dissolved over a period of time and then release the gold nanoparticle. Or you could also imagine, temperature dependent, unraveling of the polymer. That's possible too. So yeah, there are several strategies that are very well known.
Any suggestions of these polymers that you think might be useful?
That's $1 million question. So without knowing the concept, without knowing more specific details, it'll be difficult for me to say.
That's okay. Okay. Thank you very much. Okay.
You're welcome.
Thank you for that question, and thank you once again, Professor Challa. Are there any other questions? Is there any other participant who wants to raise any question?
Lairu, there was a question from Lochana. Have you asked that question?
Yes, I actually asked that question, Lochana. Okay. That was a question on spions. Yes, we went through that question.
Yes.
So looks like there are no other questions, sir, but to finish off the QA session, I would like to ask one more question from you, sir. Well, we all know that nanotechnology is a very powerful tool that we have in the present times, and we are really hopeful that nanotechnology will give us the answers to most of the questions that we have. But as a scientist who has closely worked with nanotechnology, sir, what do you envision as the future of nanotechnology? How powerful do you think is, especially in terms of eliminating the deviations that we have within the society and coming all the way from the other end of the world? I mean, do you think that nanotechnology is powerful enough to alleviate the poverty and answer almost all the questions that we have within the time to come, sir? What do you think?
Okay, very good question. Complex question to answer. Um, uh, Again, remember, there is no magic, right? There is no magic potion. Nanotechnology, nanoscience is only a tool, right? To enable us to find solutions to the complex challenges that we all face, right? Like any tool, it has its applications, it has its advantages, it has its disadvantages, right? There is no one size fits all, and nanotechnology cannot be an answer to all our problems in this world, right? But having said that, nanotechnology or nanoscience, it's basically a science. We are all scientists, and science is, should enable us to collaborate, enable us to live together, enable us to, take advantage of our combined strength, team strength, collaborative strength, partnerships. I think nanotechnology, certainly in my own experience, brought people who are so narrow, working in their own individual laboratories. I mean, I know instances where professors wouldn't talk to the neighboring professor in the same department, right? But nanotechnology really enabled us to talk to one another, not only within our department, but also across the departments, across the countries, right? That's the purpose of science and technology. And we are fortunate to be following that path. And we should never forget the greater goal, right? You guys have done an amazing job in selecting the theme for this, which is better tomorrow, right? So how can we have a better tomorrow when we do not see the most important aspect of science in general and nanoscience and nanotechnologies for us to come together, bring us together, solve the problems together, right? So that's the main purpose. And I think, like science, if you can see it properly and if you can utilize the nanoscience and nanotechnology tool rightly, that's where we would want to reach.
Thank you very much, Sant. With that question, in the absence of any further questions, I believe we have come to the end of the QA session. Indeed, it was a great pleasure interacting with you, Professor Challa, because I mean, the way that you have presented yourself and answered the questions, it's inspirational. So thank you very much for sparing your very valuable time to be here today, sir. With that, once again, I would like to remind that we have come to the end of the QA session. So thanking the speaker one more time, I would hand over the controls to Professor Rohini. Over to you, madam.
Thank you.
Yes, okay. Thank you very much, Slido. You did a wonderful job. And Professor Kumar, and it's a wonderful time that we spent with you. And it seems that we need to have more webinars with you because there are so many questions to be solved. And you showed us the importance of collaboration, and it's so fantastic. And I hope that we can have you again in somewhere during this year. It's to get some more advisors and more research avenues. And so nice. And Professor Kumar, thank you so much. And I really appreciate your time and your contribution and precious time you spent with us. And I mean, it's really happy to see you have given a positive mind for us. And it's inspirational things that you have given to our students. And you showed the importance of collaboration. It's very important. It's really important that we can see and we can get rid of the inequality. And it's thank you so much again. And now I would like to invite Dr. Gayatri Silva. She's the secretary of the SLAAS section E2, and she will deliver the formal vote of thanks note to you. Thank you so much. Over to you, Gayatri.
Yes, Professor Rohini, thanks. So hello everyone, we are at the end of today's session. And on behalf of SLAAS E2 2021 team, I would like to thank Professor Kumar Challa for taking time from your busy schedule and accepting our invitation to share your expertise and most up-to-date scientific information on modern applications and state-of-the-art techniques on nanoscience and nanotechnology.
and more specifically on how we can use these nanotechnology tools for various applications to basically address three major global challenges, as you said: inequality, biomedical, and energy challenges. And also, it is very inspiring to see how you drive continuing development of your research and innovation at Harvard University, and at the same time being engaged in R&D to ensure the delivery of research innovations to meet STEM education and research needs. So we wish you all the very best to drive your research to the next dimension in this new year, 2021. And we are eagerly waiting for the exciting scientific developments coming from your lab to successfully address these global challenges. So thanks again for honoring us with your presence. And I'm also thankful to the students, academics, and researchers for your participation, response, and cooperation to this event. So, and all these webinars, I have to say, organized by SLAAS E2 will be available on SLAAS YouTube channel. So please visit www.slaas.lk for more information. So thanks again, everyone. And over to you, Professor Rohini. So,
Thank you, Gayatri. Thank you very much, Dr. Kumar.
You're welcome. Good seeing all of you, and I'm very impressed with the wonderful work that you're all doing.
Thank you, Challa. Bye-bye.
Yeah, good seeing you.
okay, have a good day.
Bye-bye. Have a great time.
You too.
Bye.
Take care.
Bye-bye.
Keep in touch with you. Okay. Bye-bye.
Bye.
Thank you so much.